Good day, viewer, and welcome to our protein synthesis demo, presented by Deva, Rajveer, and Nisa. Before we get into our demo and even our descriptions, let's look at our table of contents. For our presentation today, we will have our glossary and introduction, we'll have transcription and its steps, and translation and its steps. Now, the glossary. To begin, let's look at protein synthesis. This is a process of creating protein molecules. In biological systems, it involves the amino acid synthesis, transcription, translation, and post-translational events. Gene expression. This refers to the synthesis of a protein based on the DNA sequence of a gene. It can also be referred to as the theory that genetic information flows from DNA to RNA to protein, and is often called the central dogma of genetics. Transcription. It is a synthesis of RNA from DNA from a DNA template. Translation. This is the synthesis of protein from an mRNA template. RNA polymerase. This is the main enzyme that catalyzes the formation of RNA from a DNA template. Promoter region. It's a sequence of nucleotides in DNA that indicate where the RNA polymerase complex should bind to initiate transcription. Precursor mRNA or pre-mRNA. It's an mRNA that has not undergone processing yet. Mature mRNA, on the other hand, has undergone processing. 5' cap. This is a modified form of a gene nucleotide added to the 5' end of the mRNA. 3' poly A tail. It's a series of nucleotides added to the 3' end of an mRNA. Splicing. In mRNA, it's a process of excising out the introns and combining in the exons. Transfer RNA, or tRNA. It's an RNA molecule that links the codons of mRNA to the corresponding amino acids for protein synthesis. Anticodon loop. This is a triplet of bases positioned at one end of the tRNA that recognizes base pairs with codons and mRNA during protein synthesis. Acceptor stem. It's the 3' end of a tRNA molecule that is the site of attachment for a particular amino acid based on the anticodon. Amino acyl tRNA synthesis. This is an enzyme responsible for the attachment of amino acids to a tRNA. Ribosome. This is a cell structure composed of the proteins and RNA that provides the site where protein synthesis occurs. Ribosomal RNA or rRNA. The RNA, this is the RNA that is associated with the proteins in a ribosome. Polyribosome. This is a structure composed of multiple ribosomes along a strand of mRNA. Start codon. A triple of these bases that specifies the first amino acid of a protein. Reading frame. This is collectively the codons of the mRNA that are read to produce an amino acid. Acid sequence. It is set by the start codon. Peptide bond. This is a covalent bond formed between two amino acids during protein synthesis. Finally, let's get into our introduction. As we have mentioned before, gene expression refers to the synthesis of protein based on the DNA sequence of a gene. The theory that genetics flows from DNA to RNA to protein is often called the central dogma of genetics. For gene expression, there are two steps that are involved. These are transcription and translation. First, let's look at our first process, which is transcription.
Transcription is the first step of the process in protein synthesis. It's the first step and in which the information in a strand of DNA is copied into a new molecule of messenger RNA. The process of transcription takes place in the cytoplasm in prokaryotes and in the nucleus in eukaryotes. In this demo, we will specifically talk about eukaryotes. Transcription uses DNA as a template to make RNA, which is the molecule. During transcription, a strand of mRNA is copied and made that is complementary to a strand of DNA. Eventually, portions of transcribed mRNA will be made into functional proteins. Transcription takes place in 3 steps. Rajveer will be explaining initiation.
Let's look at the factors associated with each step, starting with initiation. During the initiation phase, the correct position for transcription to start is selected, and the transcription machinery, which is composed of large protein and DNA complexes, is assembled. For each gene, only one strand of the double-stranded DNA molecule is transcribed. This strand is called the antisense strand or the template strand. The other strand, which is not transcribed, is called the sense strand or coding strand. It has the same sequence as the product mRNA and with thymines instead of uracil. And in one single DNA molecule, either strand could serve as the sense strand for different genes. The main enzymes that catalyze the synthesis of RNA are a group of enzymes called RNA polymerase. In eukaryotes, each RNA polymerase has a specific function. Transcription begins when RNA polymerase binds tightly to a promoter region on the DNA. This region contains special sequences of nucleotides. This signals the DNA to unwind, so the enzyme can read the bases in one of the DNA strands. The enzyme is now ready to make a strand of mRNA with a complementary sequence of bases.
All right, so the next step is elongation. The RNA polymerase complex works its way along the DNA molecule, synthesizing a strand of mRNA that is complementary to the template strand of DNA. In the mRNA strand, however, the thymine is replaced with uracil. Like DNA polymerase, RNA polymerases work in the 5' to 3' direction, adding each new nucleotide to the 3-OH group of the previous nucleotide. RNA polymerases transcribe only one strand of the template DNA, so there's no need for Okazaki fragments as in DNA replication. As soon as the RNA polymerase complex starts to move along the DNA, a second RNA polymerase complex can bind to the promoter region and start to synthesize another mRNA molecule. This means that hundreds of copies of mRNA molecules can be made from one gene at one time. Also, RNA polymerases catalyze the synthesis of mRNA at a much faster rate than DNA polymerase catalyzes the synthesis of DNA. This is mainly because the RNA polymerase complex does not have proofreading function. An error in transcription would only result in an error in one protein molecule and not in the genetic makeup of an organism. Being able to synthesize more mRNA in a given amount of time is a greater advantage than minimizing sequence errors. It is the addition of nucleotides to the mRNA strand. RNA polymerase reads the unwound DNA strand and builds the mRNA molecule using complementary base pairs during this. During this process, an adenine in the DNA binds to an uracil in the RNA.
For the termination phase, specific nucleotide sequences in the template DNA serve as a signal to stop transcription. When the RNA polymerase complexes reach the signal, they detach from the DNA strand, and the new mRNA strand is released from the transcription assembly. And then the DNA double helix reforms. So here you can see the RNA detaching from the DNA strand, and the DNA double helix is starting to reform.
Yeah, good job. Anyways,
Thank you.
intermission.
Intermission.
You had a
Let's
busy-
look at our post-transcriptional processes. After transcription, eukaryotic pre-mRNAs must undergo several processing steps before they can be translated. Eukaryotic and prokaryotic tRNAs and rRNAs also undergo processes before they can function as components in the protein synthesis machinery. A cap is added to the 5' end of the growing transcript by phosphate linkage. It's called the 5' cap. This addition protects the mRNA from degradation. In addition, factors involved in protein synthesis recognize the cap to help initiate translation by ribosomes. Once elongation is complete, an enzyme called a poly-A polymerase adds a string of approximately 200 A residues called a poly-A tail to the pre-mRNA. This modification further protects the pre-mRNA from degradation and signals the export of the cellular factors that the transcript needs to the cytoplasm. Pre-mRNA splicing. Eukaryotic genes are composed of exons, which correspond to protein coding sequences. Exon signifies that they are expressed, and intervening sequences called introns. Intron denotes their intervening role, which are removed from the pre-mRNA during processing. Intron sequences in the mRNA do not encode functional proteins, so they are pretty much useless. All the pre-mRNA's introns must be completely and precisely removed before protein synthesis. It is the process urged by even a single nucleotide. If the process errors by even a single nucleotide, the reading frame of the rejoined exons would shift, and the resulting protein will be dysfunctional. The process of removing the introns and extrons and reconnecting the exons is called splicing. Here we can see the 5' cap added to our mRNA strand and the poly-A tail. And on the next slide, we can see how the introns are removed and the exons are joined together to give a continuous movement of the mRNA, making it a mature mRNA strand.
Yeah, man. All right, so now let's get into the second part of protein synthesis, where the protein is actually made. It's called translation. The process of translation or protein synthesis involves the decoding of a mRNA message into a polypeptide product. Amino acids are covalently strung together by interlinking peptide bonds. Each individual amino acid has an amino group and a carboxyl group. Polypeptides are formed when the amino group of one amino acid forms an amide bond with the carboxyl group of another amino acid. This reaction is catalyzed by ribosomes and generates one water molecule. So, ribosomes. A ribosome is a complex macromolecule composed of structural and catalytic RNAs and many distinct polypeptides. Ribosomes exist in the cytoplasm in prokaryotes and in the cytoplasm and rough endoplasmic reticulum in eukaryotes. Ribosomes are made up of two subunits. In E. coli, the small subunit is described as 30S and the large subunit is 50S, for a total of 70S. Mammalian ribosomes have a small 40S and subunit and a large 60S subunit for a total of 80S. The small subunit is responsible for binding the mRNA template, whereas the large subunit sequentially binds tRNAs. And here is the structure of the ribosome. So here is a tRNA structure, and basically the tRNA, also known as the transfer RNA, is a structural RNA molecule that were transcribed from genes by RNA polymerase III. They serve as adapters. Specific tRNA molecules bind to sequences on the mRNA template and add the corresponding amino acid polypeptide chain. Therefore, tRNAs are the molecules that actually translate the language of RNA into the language of proteins. Here's the anticodon, which binds to the mRNA codon. And yeah. So now we're going to be discussing the different steps in translation. And again, it's initiation, elongation, and termination.
In the initiation phase, all the translation components come together. Proteins called initiation factors assemble the small ribosomal subunit mRNA, initiator tRNA, and the large ribosomal subunit for the start of the protein synthesis. The small ribosomal subunit attaches to the mRNA near the start codon AUG. The tRNA that binds to the codon is the initiator tRNA with its anticodon. In prokaryotes, this tRNA carries a derivative of methionine. In eukaryotes, methionine is used. Then a large ribosomal subunit joins to form the active ribosome. The start codon sets the reading frame for the gene. The reading frame established how the subsequent codons in the sequence will be read. The three binding sites for the tRNA are the P peptide site, A amino acid site, and E exit site. During protein synthesis, the P site contains the tRNA with the growing polypeptide attached to it. The A site contains the tRNA with the next amino acid to be added to the polypeptide chain, and the unchanged tRNA, which no longer has an amino acid attached to it, is ejected at the E site. At initiation, the initiator tRNA binds to the P site.
All right, so now for elongation. In the elongation phase, protein synthesis occurs. The polypeptide becomes longer, one amino acid at a time. In addition to tRNAs, elongation requires elongation factors, which enable tRNA anticodons to bind to mRNA codons, like I said before. Once the initiator tRNA is bound to the ribosome, the A site is occupied. Let's find a picture of this. All right, we'll look at the picture after. Anyways, so basically, in addition to tRNAs, elongation requires elongation factors, which enable tRNA anticodons to bind to mRNA codons. Once the initiator tRNA is bound to the ribosome, the A site, or the amino acid site, is occupied by the tRNA with an anticodon that base pairs with the second mRNA codon, and that carries the second amino acid of the protein being synthesized. A peptide bond forms between the first and second amino acids, and the resulting dipeptide is attached to the tRNA at the A site. The mRNA moves along by one codon, and this complex becomes associated with a P site of the ribosomes. The sequential addition of amino acids during elongation is a cycle of 4 steps that is rapidly repeated. First, a tRNA with an attached polypeptide is the P site, and the tRNA carrying the next amino acid enters the A site. Next, the polypeptide chain is transferred to the amino acid of the tRNA in the A site. This makes the polypeptide chain 1 amino acid longer than before. Last, the mRNA moves forward by 1 codon, and the polypeptide bearing tRNA is now at the ribosome P site. The uncharged tRNA exits. The new codon is at the A site and can receive the next complementary tRNA carrying the next amino acid of the polypeptide.
Now, to really end this entire process of protein synthesis, we have the termination stage. The termination stage begins when a stop codon on the mRNA is reached. The polypeptide of the components of the translation machinery are then separated. A protein called a release factor cleaves or cuts the polypeptide from the last tRNA. The polypeptide is then released and will eventually fold into its 3-dimensional shape, ready to carry out its cellular activities. Now we have the images to show our entire stages of translation, from the initiation phase to the elongation phase, where we can see the proteins being created, and lastly, our termination phase, where the protein is finally cut free and is free to carry out its cellular activities. The newly formed polypeptide chain can now undergo its several modifications before becoming a fully functional protein. The proteins have a variety of functions, and some will then be used in the cellular membrane. Conclusion. While others remain in the cytoplasm or will be transported out of the cell, many copies of the protein can then be made from just one mRNA molecule. This is because several ribosomes can translate the same mRNA molecule at the same time. These clusters of ribosomes that translate a single mRNA sequence are called polyribosomes or polysomes. Thank you so much. This has been our DNA. This has been our protein synthesis DNA demo script. Thank you.
Yeah, man.
Yo, stop recording.
Okay, and let's